Hi, hello everyone. I'm very thankful to be here with you all today. My name is Jess Sterling and I'm a third year PhD student at UT Austin. I'm one of Justin's grad students. And today I'm going to be talking to you all about some research that we actually recently published over the summer. So the primary purpose of our experiments and stuff for this particular project was looking at primary microbial succession. So what is that? Essentially, how I like to view it is the conquering of a new kingdom. So what it's defined as is the conquering of sterile lands. It's the first microbial organisms that show up to a new environment. So these ecosystems can be terrestrial or aquatic, but either way, microbes are almost always going to show up and start fighting. And so I kind of like to view this as a tiny kind of microbial war that goes on, and essentially different species of microbes start to show up in these new ecosystems, and they start battling to determine who's going to be the dominant species in that area. So like most wars, there's victors and there's also losers. And what this looks like within these ecosystems is that some of these species will make up the vast majority of the population of microbes within that community, and then some will disappear entirely. So why does this matter? Why are we interested in microbial succession in the first place? For one, as some of the talks before me talked about, these microbes are actually critical for the food web in these alkaline pool systems, as well as nutrient cycling. So things like the nitrogen cycle and the carbon cycle, which happens at the global scale, also happens at a local scale within these new environments. And especially within aquatic environments, if these microbes don't show up and start the process of fixating nitrogen, etc., you can actually have more complex life live in those ecosystems. And lastly, it establishes a level of stability, so having stable levels of these nutrients, as well as being able to remove toxins and waste products from more advanced life forms, is critical for this process. So the environment that we're interested in studying, as you can imagine, sterile environments don't just pop up all that often today, but volcanic activity can lead to the creation of some of these environments. So some work has been looked at the microbes and how they establish themselves in terrestrial environments in these lava ecosystems, but not so much within the marine environments. So as far as our site, we had a unique opportunity. In 2018, there was a lava flow here on the Big Island, and it led to some changing of the geology of the coastlines here, and also led to the creation of a couple new alkaline pools, which hadn't happened for quite some time. So here's kind of a photo. Here's my PI Justin and our collaborator Troy at one of these new ponds that was formed from this lava flow. And one thing to note, as kind of mentioned earlier, there are these microbial mats that form in these ponds. So up top is an established pond in the A section with the orange cyanobacterial crust that was mentioned earlier. And at the bottom is an example of one of these new ponds. It looks more kind of like the algae mud type of environment. So what do we currently know about the microbial communities within these ecosystems? For one, we know that these ecosystems are largely unique. Even nearby ponds have very distinct microbial communities from one another. And secondly, within these established ponds, these communities tend to remain pretty stable and don't have a lot of fluctuation from year to year. And we know this thanks to some research from Stephanie Hoffman's lab. And now we wanted to look into these ponds and had a couple of hypotheses, the main thing being that we figured that these alkaline ponds, these newly formed ones, their microbial community would start trending towards and becoming more like established ponds with time. As far as the objective, we had two main things that we were trying to accomplish. The first was to identify the microbial communities, so pretty much what species are present within these new ponds, what does that community look like, who has the majority makeup of these populations, etc. And two, we wanted to track the changes in these compositions over time. So was the composition same at the beginning of this time points versus the end of our experiments? So I've broken up the methodology into 4 central parts. And I'm going to go through each of them one at a time, starting with the physiochemical aspect of things. So this was pretty much, we wanted to look at things like the oxygen concentration, salinity, what kind of compounds were present, etc. And the reason why we did this is because this determines the habitability for different microbes. So pretty much these factors determine which microbes can and cannot thrive within this environment to begin with. So we wanted to make sure we understood what was going on. Secondly, we did some sampling of the benthic microbial communities, and I say we, but I actually didn't get to sample. Troy did a lot of the sampling for us, so we're very thankful for that. And so we looked at these 3 new ponds at 3 different dates across the course of a year, and did, of course, repetitions at each time and preserved these samples for analysis. And so in terms of the DNA profiling and assessment, we extracted our DNA from these samples and then amplified the 16S and 18S regions of the genome. So essentially what these are about 100 base pair long segments that are hyper-conserved and hyper-variable. So they're pretty standard practice to use these portions of the genome to identify what species are present within a sample. And mostly why we use these specific regions is we wanted to compare these new ponds to the pre-existing established ponds, and the research that had been done used these same regions, so we wanted to maintain them. And then lastly, we wanted to check the composition of these microbes over time. So essentially, how do these communities change over the course of a year? So now I'm going to go into some of the results and analysis. Starting with the physiochemical side of things, I want to focus mostly on the parts of the environment that seem to vary from established ponds. The first being temperature. The temperature was very warm compared to established ponds, so they were as high as 44 degrees Celsius and ranged closer to the low 40s across the three ponds at all three time points. And these normal established ponds have temperatures closer to the mid-30s. The silicate levels were also pretty high compared to established ponds as well, so we found that to be interesting. And then lastly, the oxygen concentration was normal in the beginning during our first sample, but then increased to almost 4 times the normal amount of oxygen in later samples. And lastly, this was actually pretty stable and pretty similar to the established ponds, the salinity, but it was variable, which is pretty common within these ecosystems, so there was nothing really major there. So as far as our results, looking at the community analysis and tracking the changes over time, so the red star indicates where these new ponds were that we studied, and these boxes indicate other ponds that we use as a reference points. So kind of phenotypically, scientists will classify these ponds based on how the microbial mat appears. So there's the orange crust, the algae, and the mud, and so we wanted to see how they compared to some of these established ponds. So here is an NMDS plot, which essentially categorizes and classifies community makeup based on diversity and what species are actually present. So 2 dots, when they're close together, that means their communities are very similar. And as you can see, the orange crust communities had a tendency to cluster towards each other, but individual ponds actually still very strongly correlated to itself. Same thing with the mud and algae ponds. And so now here, this is the data we generated with the new ponds. And as you can see, they're very different from the established ponds. They're a lot more diverse when we're looking at the bacteria. There's nearly 3 times as many species present. And then also we tracked it over time. So as you can see, some of these boxes get darker and is indicative of a later time point. So the darker boxes actually have a tendency to move away from these established ponds. And we were actually expecting the opposite trend to happen, so we found that to be pretty fascinating. Not sure why that happened, but it did. And here's kind of a zoomed in look at the actual composition of these microbial communities here in the established ponds. As you can see, they're mostly made up of cyanobacteria and protobacteria and don't have a lot of diversity. But with these new ponds, they are a lot more diverse, and even though they're high in cyanobacteria, it makes up a significantly less portion of the population. And there are also some other bacteria that are present that, like these two listed here, Chloroflexi, and they actually made up about 10% of the microbes in these established or new ponds, but they made up a fraction of a single percent in the established ponds. So there was a lot of difference. And then lastly, we wanted to track the changes over time. So here's a zoom in looking at the composition of the bacteria from pond 1 across 3 time points over the course of the year. And as you can see, the changing of the bars indicates the composition of these communities actually changed pretty significantly from each of the 3 time points, which suggests that they haven't reached a point of fixation or stability like is present in some of the established ponds. Okay, and secondly, we looked at the eukaryotic composition. Again, you can see that the orange crust communities and the mud algae communities tend to cluster towards each other. But when you looked at the new ponds, for one, they had significantly less diversity. There's nearly 7 times as many types of species within the established ponds than compared to the new ponds. They also seem to be very distinct communities, even between ponds. As you can see, they're clustering away from each other. And then if you're looking at the actual composition makeup of these, you can see that there's a lot of diversity amongst eukaryotes within the established ponds. As you know, we talked about today, different things like mollusks and the shrimp that are present make up a high diversity. But within these new ponds, they're very low in diversity in terms of the eukaryotes. They are missing a lot of algae. There's next to no presence of the shrimp or mollusks, which are pretty high in the established ponds. And then similarly, like with the bacteria, we did see significant differences from the 3 time points of the composition of eukaryotes. And so we kind of expected this, as the microbial community still hasn't established itself, which maintains the stability within these environments. The eukaryotes still haven't quite found their way into these ponds or established themselves nearly as well as in the established ponds. So as far as conclusions, the young anchialine habitats have very distinct microbial communities, we found, and we actually saw that they were trending away from the established ponds. So this suggests that they might be able to facilitate unique taxa that some of the other anchialine ponds might not be able to. And secondly, we did notice that there is high levels of fluctuation within the microbial community, within these new ponds. And unlike the established ponds, there's not nearly as much stability yet. And we may have captured some fine tuning more so when it comes to this, because we didn't start taking samples from the new ponds until about a year after these ponds had formed. So we figured that if we would have taken samples a lot earlier, we might have seen even larger fluctuations happen over time. And so lastly, I kind of want to talk about some of the conservation implications of this research. For one, as I mentioned, the new ponds are very unique in their microbial community makeup, and since they're trending away from the established ponds, it could be that they're able to sustain unique life forms, and because of that, we might want to use special conservative concern when considering these younger habitats as a protected ecosystem that might be able to hold and sustain very unique life. And then lastly, I'd like to thank all of you, as well as all of our collaborators, our funding sources, and this research was actually published earlier this summer in Integrative and Comparative Biology. So if you're interested in learning more, feel free to peruse that. And now I'll take any questions.
All right, Jess, time for 1 or 2 questions.
These new pools that are forming down in Kuna, I assume they're warmer because they still molten lava. Are any of these microbes examples of extreme ponds?
Not the ones that are making up the predominant proportion of the microbes in the community, not necessarily. You're still seeing some of the ones that are present within the normal ponds, not necessarily in the extremophiles, just in higher proportions.
Do you expect a succession over time as the pools cool while the lava?
Yeah, of course, because the temperature we're suspecting does have an impact on which microbes are able to thrive in these ecosystems. And even now, like at the beginning, the temperatures were actually lower than we saw towards the end. So there is fluctuations in the temperature, and we suspect that eventually the temperatures will stabilize, and that'll probably impact the makeup as well. So it might take a while for these microbial communities to actually reach a stable place.
Hi, that was really a great talk. Have you guys looked at the possibility that land use might be affecting the microbial communities? Just because I know that some of the ponds are kind of embedded in pretty heavily used areas. Groundwater might be containing nutrients and so forth. And I've just, with all that diversity, I wonder if there's an effect there.
Yeah, so that's a good point. Within these newer ecosystems, that's where we did our studies. As far as the established ponds, I'm sure there's probably some impact, but that's not anything that we particularly focused on. But I imagine it would definitely impact, because like I said, a lot of those geochemical aspects of those ponds have an impact on what kind of microbes can even sustain themselves. Like certain metabolism, scientists can determine that they are not able to occur based on different parameters of the water quality. So it is likely that any kind of human effect would limit what kind of microbes could exist in those ecosystems.
We've heard today that a lot of these anchialine ponds are interconnected and the shrimp go back and forth. And so at what point do you expect these new ones to start to interconnect with existing ones?
We're not sure, but we think one of the biggest hindering factors is the temperature right now.
Right.
We think that the temperature might be a little too high for the shrimp to actually live comfortably in.
Right.
So until the temperature goes down, we don't think that they'll actually start to have a local ecosystem within these new ponds. Yeah.
You see now when they do. Is that like 5 years away, 10 years away?
I have no idea.
Actually, USGS HBO just had an article a week or two ago and was talking about the rate of cooling as a function of the thickness of the lava flow in those areas. And so, so if you want to read it, I think it's online on the HBO. And so there's a lot of variation in that particular flow and when they think it's going to get cool.